landscape, and before I get going, I just want to say that it's all a major collaboration with Brenda Andrews, who's the director of our institute here at the Donnelly Center. And I'm going to try and explain why this graph is a model of the cell. That's basically what I'm going to try and tell you guys. And yeah, it could be anything, right? But here's the model that we have so far. And when you think about it, we know a lot about the parts of the cell, but we really don't have a good model for how they're integrated together. And functional genomics, systems biology tries to do that in a quantitative way. It's not that we're very good at it at this stage, but it's sort of the beginnings of it, and our project has kind of contributed to this. So I'm going to talk about the budding yeast, which a bunch of us work on here. Georgiana and David are leaders in the field, and it's a eukaryotic cell, so it's a billion years further evolved beyond prokaryotes, and it's got a nucleus and a mitochondria with its own DNA. It's got all the goodies that we find in human cells, and so it's been a major model organism. And in fact, there's 1,000 yeast labs around the world, and we all feed data into one big common database that is put together at Stanford by SGD. So yeast has 6,000 genes and 16 pairs of chromosomes, and you compare that to a human has 20,000 genes, 23 pairs of chromosomes. So it's a pretty sweet little cell. And the yeast community, this international community, there's labs all over Europe, major labs in Japan, and of course in North America, and it's expanding to China and India rapidly as we speak. Not in Russia. There's nothing in Russia. I think that's weird. But anyway, the goal is to figure out. So we've got a graduate student. There's 6 graduate students each lab. We've got a graduate student for every single yeast gene, right? And you'd think that we can figure out how this cell works. We can model it, but we're so far from it because we have to figure out how they're all connected to one another and how they're talking to each other and the dynamics of it all. And that is way beyond the capability of the yeast community, but any community studying any form of life. But just to give you a view of that, here's 16 to 16 chromosomes. Each line represents a gene. So these genes are just lined up one after another on each chromosome. And one thing you can do with these, because it's so easy to manipulate this organism genetically, partly through homologous recombination, is we can go into a diploid, so it has 2 copies of every chromosome, a diploid organism, and delete 1 copy of each gene. And then the beauty of yeast is you can take it and convert it from a diploid to a haploid, where it only has 1 copy of each chromosome. And you can, under those conditions, ask whether that gene is required for life. And so the yeast community did this, deleted each of the 6,000 genes, one by one in a diploid, converted it to a haploid, and asked whether the gene was required for life. And 1,000 of the 6,000 genes, when deleted individually, is required for life. 5,000, you delete them in the cell, basically everyone knows it's happened. Okay. And this is true not just in yeast, it's true in all forms of life, basically, any sophisticated form. So in E. coli, in yeast, in worms and flies and mice, and in human cells, only a fraction of the genes are required for the basic cell division and replication of chromosomes. Most of the genes appear to be functionally redundant, okay, so that you can delete them, and they must be doing something because they're highly conserved, but it looks like there's a backup plan for whatever the role of this is. So a nice example is DNA repair. So there's many pathways that control DNA repair, different pathways, and they're required under particular conditions. But in a laboratory setting, you can remove non-homologous end joining, and the cell's perfectly fine. It can repair its DNA through homologous recombination, for example. But if you remove both those pathways, homologous recombination, non-homologous end joining, then the cell has a problem. So we think many of the reasons these cells aren't required for viability is because the cell is wired with backup systems, and so that the analogy is it's like a finely tuned jet engine where if something goes wrong, we've got another way to keep the plane up in the air. So
why maybe it's because of people in women, why would the cell work against the 1,000 remaining genes and have backup?
That's a good question. I can't figure that out, except to say that it wants to streamline everything as much as possible, but still, then why wouldn't there be a backup pathway for that thing? Yeah, and there's really only one proteasome. It doesn't make another copy of all those genes and another. There's other ways autophagy thinks to degrade proteins, but the major essential complexes are things like the ribosome, the proteasome, and the whatever.
Just make sure you're not telling us that you had a yeast cell where you deleted 5,000 genes and the remaining thing is alive, right?
Yeah. No, but that's not like so. Craig Venter tried to make the minimal genome, right? And basically, if you read their paper, they tried to take the equivalent of these 1,000 genes, and see if it would work. And then, they're really surprised when it didn't work. So yeah. You won't even give me any feedback, like. Yeah. He did bacteria, but they basically their idea was this, and then if you read their paper, they're really surprised to rediscover synthetic lethality.
So maybe backup is not the right way to think about it.
Yeah, I don't think it's the best way to think about it. And it's way more complicated than just one pathway backing another one out. Like, so when I show you, so it's just a very simple beginning thought that's not correct.
Can I just say something that, for example, some of the genes become essential once you give them other conditions, for example, stress. So now they become essential. essential. So they're all essential. The viability are essential. So there are more essential genes than other things.
Yes.
So it's actually not really linear, but still why they don't have a backup.
Yeah.
There are some backups, right? No, and some not.
Yeah, I mean, it's complicated, right?
Yes.
Yeah. Okay, so anyway, so here's what we're going to do. We're going to explore gene function through this genetic interactions, and this is a term that does not mean it does not mean interaction physically. Okay, it just means like some kind of more like a mental interaction, right? Sex. No, I don't know what it means. And but here's a definition that a genetic interaction occurs when mutations in two. We'll just focus on two right now, but it could be more. Two different genes combine to generate an unexpected double mutant phenotype. Okay, so if I have mutant A and I know what it does to the cell, and I have mutant B and I know what it does to the cell, when I put those two together, I expect to see both things happening, right? That's the additive effect of the double mutant. But we call it genetic interaction when I take this and this and put together, then something crazy happens, right? Something unexpected. And so the guy that we all know and love that articulated this to the yeast community was this guy, David Botstein, and he really did articulate this when you go back and look at the literature, and which is kind of surprising because we think that the yeast community thinks it's been looking at genetic interactions for years and years and years, but it wasn't actually. I'm almost going to graduate from my PhD at this point, and this paper from famous Peter Novik and Botstein comes out, and they're looking for suppressors of the genetic interaction of yeast actin mutations, and so and this is a yeast cell stained with mentose actin with rhodamine. Yeah, maybe turn that. Yeah, that's a good picture. Yeah, I know. Yeah, so of course David is the yeast actin expert. But look at that mother cell, the daughter cell. We've got these patches here. David's going to talk about they're all called endocytosis, and then these cables that are tracked from myosin motors that built by glycogen. And yeast only has one actin gene. Okay, so this makes it, and it's essentially mutated phenotype of these guys. The cell can't morph any shape properly, and it can't divide. But and if you have a worm, a nematode, they have lots of different actin genes, and so you can't really do this type of experiment with a worm. So what Novik and Botstein did was they mutagenized the actin gene, and this -1 means it's a particular allele, so some particular mutation, and they isolated a mutant that was alive at 23 degrees and dies at 37. Okay, so this is a temperature-sensitive mutant, and geese and fly geneticists use this to study essential genes in a conditional way, so we can grow them at a permissive temperature and then shift the restrictive temperature and see what happens, reveal the essential phenotype. And one way to think about it, but there's lots of different things that happen. The protein might not be made at the restrictive temperature, it might be degraded, but the classic idea is that you have a mutation in the polypeptide chain that allows it to fold normally at the